Bueno, cuando quieras, Juan.
Sí se está compartiendo, ¿no?
Sí.
Okay. Today I'm going to show you this paper called One Chip Analysis of Carbon Dots Effect on Yeast Replicative Lifespan. So I was searching for different designs of chips to perform live cells culture, and I found this, and I'm going to show you now. This is the design, and they use this gradient to dilute a higher concentration of carbon dots. Well, these carbon dots are promising nanomaterials for biosensing, bioimaging, and drug delivery. Due to their large surface area, excellent optical properties, and thermal and chemical stability. But the biosafety of these these carbon dots is still understood. They present in this work a gradient network generator, a microfluidic device for dose-dependent testing of toxicity of carbon dots to a unicellular eukaryotic model, in this case, Pichia pastoris. So to understand the toxicity mechanism, they study the binding of these carbon dots to the yeast Pichia pastoris, and found that the increasing interactions of these carbon dots with the cell's surface area at carbon dots is in larger concentrations. And they found in this study that the utility of this gradient generator microfluidic device as a convenient tool to for toxicity assessment of nanomaterials at a cellular level. The concentration gradient generator that they call CGG is a microfluidic device that offer a convenient platform to evaluate the toxicity of the carbon dots in the cells. They applied a tree-network microfluidic device for dose-dependent testing of these carbon dots effects on various properties of unicellular eukaryotic model. They synthesize a blue emitting carbon dots, and after that they characterize using electron microscopy, dynamic light scattering, and spectrophotometry. And I found this very interesting. They optimized the geometry of the cell microchamber integrated with a tree-shaped microchannel network to decrease the potential effects of shear stress on budding yeast. And they measured the proliferation rate and morphology of the yeast cells in the presence of different concentrations of the carbon dots and the binding of these carbon dots in the cell surface. And they found that the large surface area to volume ratios and the low flow shear stress in microchannels lead to the microenvironmental homogeneity. Well, in the methods, as I told you, they synthesize the carbon dots. After that, they make the microfluidic device. This is the design. The first part is the concentration gradient generator module and the cell chamber module. And they test the microfluidic device operation to perform these assays. In the results, the characterization of the carbon dots, they used these images with the temp, and after that they also perform a simulation of this mixing efficiency and the shear stress in the microfluidic device. In figure 3, in this one, they show the mixing of the efficiency at the endpoint of the gradient generation module, and the mixing efficiency decreased by the increase in the flow rate due to reduce time diffusion, but remained as high as almost 97%, as you see in the graphic. And this demonstrates that the effectiveness of the tree network design for mixing the dilutions. Okay. And this the fluid shear stress in the microchannel can disturb the budding process in the yeast cells and cause the cell morphology variation. The chamber model of this microfluidic device was designed to confine the cells to a no-flow condition and avoid this shear stress on the yeast cells. The closure of this cell chamber causes the maximum amount of fluid to flow through the main channels and does not perfuse into the chamber due to the convection transport, which provides a condition to isolate the cell's proliferation chamber from bulk fluid flow. And in the part C of this figure, as you can see, the image and of this part of the design. The flow velocity of 400 micrometers per second in the microchannels decreased to approximately 0 in the cell chamber. I don't know, I think it's a heat map or something like that in this simulation. So a static growth condition is formed in microchamber to facilitate a suitable condition for budding yeast cells. Okay, yeah. In this situation, the exchange between the rectangular chamber cell is supported by mainly by diffuse transport of nutrients into the chambers, which is enough to refreshment the yeast and in this microenvironment. And this one is I found very interesting. They studied the growth rate and the morphology of the yeast cells under different conditions. They use light micrographs of these cell culture chambers between 0 to 25 micrograms per milliliter, and the concentration of these carbon dots after 5 hours of incubation are shown in this figure. As you can see, the increasing of these carbon dots concentration, those dependently decrease the number of the yeast cells, and decrease in the cell in the cell number was fairly linear, and the largest concentration of these carbon dots from 25 to is like 94% fewer cells than in the control condition. And to see the interactions of yeast cells with these carbon dots, they study the binding of these carbon dots to the surface of the cells. And in the figure 7, this one, shows the carbon dots intensity emission at 4060 nanometers, and the increasing the carbon dots concentration also increase the emitted light intensity, indicating that the binding of these carbon dots to the surface of the yeast cells. These carbon dots binding to the surface of yeast cells may induce toxicity. So they simulate the they think that the carbon dots simulate the production of these stimulate the reproduction of these intracellular reactive oxygen species, ROS, beyond the capacity of the cells to neutralize them. And this could be the responsible for the growth inhibition with the binding of carbon dots at higher concentrations. And they conclude that the interaction of these carbon dots with the yeast cell surface increased approximately linearly with increase of in the carbon dots concentrations, and this potentially inhibits the growth of the yeast cells through the stimulation of cells to produce these ROS at toxic levels. Thank you.
Thank you, Juan. Do you have any questions? If not, we can follow with Andrea, no?
Yes.
Thank you,
Hello,
Andrea.
everyone. Can you see my screen?
Yes, Andrea.
Okay. Thanks, Cami. Well, today I will talk about this article called C. elegans behavioral assay distinguishes early stage prostate cancer patients' urine from controls. To begin with, detection of these cancer-related metabolites or volatile compounds in blood and urine would provide an ideal means of non-invasive early cancer detection. Yet chemical analyses have been unsuccessful in determining metabolite profiles that consistently distinguish early cancer from healthy biofluids. In the early stage of malignancy, there is evidence that cancer patients emit odors that can be accurately detected by canine and urine olfaction. C. elegans is commonly used as an experimental model in neurobiology because of this simple nervous system, which is primarily devoted to chemosensation. So, using a simple assay setup, previous assays have shown that C. elegans can detect multiple types of cancer. In this work, they apply Chemotactic assay to urine samples from patients with prostate cancer and compare the behavioral response to benign controls. Okay. The current gold standard biomarker for non-invasive prostate cancer detection is this prostate-specific antigen, or PSA, which is excreted by both malignant as well as non-malignant epithelial cells, and can therefore be present in the serum of patients with benign diseases such as prostatitis and prostatic hyperplasia. Now the court included urine samples from prostate cancer cases, control benign patients, and control negative screen patients. They use young adult stage nematodes for all chemotactic assays. Now, to determine if C. elegans displayed a behavioral response to candidate to volatile compounds, they did chemotaxis assays using dilute 2-octanol or pentanol as test samples. And C. elegans demonstrated overall attractions to 2-octanol at these two concentrations, and to pentanol, it was only at the highest concentrations. Then they perform urine samples among the 5 dilutions you can see in this part. And the discriminations of cancer patients' urine from controls in preliminary assays as defined by number of samples with overall behavioral attractions for chemotaxis index towards cancer samples and repulsion. Here you can see the chemotaxis index, positive values and negative values. And when is a positive value, it means that exists attraction to the volatile compound. So this urine dilution was used for all subsequent chemotaxis assay on patients' samples. They observed both attractive and repulsive patients' urine samples within the 3 groups. And here you can see all the samples and the values. And in this figure, after chemotaxis index for each patient were average, and urine from cancer patients elicited generally positive average to chemotaxis index compared to urine from benign and negative screen individuals. Here you can see that the positive values are in the samples of patients with cancer. And here, then they found that prostate cancer urine samples in chemotaxis index were significantly higher compared to benign negative screening individuals when all technical replicates were considered for each patient sample. So to calculate sensitivity and specificity, they use classifications and determine by pathology. True positive were considered to be individuals with a confirmed case of cancer following a biopsy, while true negatives were considered to be individuals who either had a negative biopsy or were deemed to predict cancer status. They use a model using Chemotactic index to classify cancer and or to classify patients with cancer and classify patients with cancer-free. Okay. With this model, they consider Chemotactic index and other values like prostate size or these antigens, these biomarkers. And they did a relation between these values, and of this manner, they were able to increase the specificity to 85%. And it means the combined model accuracy improved to more of 80%. In conclusion, these results support previous work that animal olfactory response could be a useful tool to discriminate cancer stage. And finally, I want to show you the scheme in agar plate to do a Chemotactic assay. I like this work because I want use the chips for do this assay. Well, that is all. Thank you.
Thank you, Andrea. Do you have any question for her? We have in the list Fernanda, but Fernanda is not in the meeting. Do you want to speak anybody, or we can finish here? Well, if no, there isn't any more presentation, I think we can finish the meeting. Thank you very much. We see us together in March. Well, bye-bye.
Okay.
Bye,
Thank you, Cami, for all.
Gracias. Bye, grabar.